Okay, so it's sort of the first day of the conference. So it all kind of kicks off this evening. I just got back from the NeuroMatch session this morning where we just matched up with some other conference attendees based on our interests, and we chat and kind of get to know each other. And now I'm just going through the abstracts. I've just gone through them, highlighted some of the interesting ones, the ones that look particularly interesting. And I thought I'd just go through some of these right now just to kind of prepare for the poster session this evening. And actually, the first session is going to be Lars Chitka talking about the mind of the bee. So I think that's quite in line with my hope earlier that there's going to be some insights from the insect brain. So I think we're going to be seeing that a little bit later today. But yeah, let's go. So after that, there's going to be a poster session. So within the app here, the Hoover app, you can kind of pick the posters you're particularly interested in. So you can go and visit them later. So I thought I'd go through these right now. The one problem is that ideally I'd be doing this with the PDF, so I can actually point to things, highlight things. I don't think they have the PDF available just yet, so it's going to be a little bit, there's not going to be a pointer on the screen, so it's going to be, so you won't quite know what I'm pointing to, but hopefully it'll be easy enough to understand. So yeah, so the very first one, and these aren't ordered, they're just ordered in the numbering that they've been assigned in the conference. So that's the order we'll go through them. It's not by order of preference or anything like that. Okay, so the very first one is distinct excitatory and inhibitory connectivity structures, control dynamics and computational capabilities of recurrent networks. That sounds quite interesting. So let's see what that is about. And if we load it up, okay, so yeah, so this notion connectivity is far from random. Higher connection probably between similarly tuned neurons across cortical layers. In theoretical studies, have been shown to generate rich dynamics and spiking networks, while the connectivity of inhibitor neurons is usually less specific than that of excitatory neurons. So they have kind of a more, maybe kind of a global connectivity, at least within their nearby circuits. So several studies have found different degrees of inhibitory specificity and diverse connectivity patterns, whose impact on population dynamics and computational implications are not fully understood. So that's kind of interesting because, for example, in the inference circuits that I work with, the connectivity, the inhibitory units, which we think are the granule cells, they embody the causes, the odor causes that we think have caused the receptor activations. And their connectivity is certainly not random. In fact, it reflects the amount that we expect each receptor type to be excited by the cause that a particular inhibitory cell is representing. So at least within the limitation of those models, we certainly don't expect connectivity to be, inhibitory connectivity to be random. But let's say what they say. So inhibitory specific and diverse connectivity patterns whose impact on, population dynamics are not fully understood. Now here we link the presence of inhomogeneous, so I guess non-random, neuron type specific clusters with complex dynamics that are associated with optimal performance in computational tasks. So the presence of inhomogeneous localized neuron type specific clusters, with complex dynamics. That's going to need some unpacking. Okay, so now they're going to use reservoir computing. So that's generally the notion that if you have pools of neurons that are connected to each other with some excitation to kind of keep things, keep the reservoir active and keep neurons coming in and out of activation, but not too much excitation, otherwise the system would become epileptic. And perhaps, I guess, in this setting also balanced by inhibition to kind of keep things roiling to some extent. That gives you lots of computational primitives. That means, time series of activities of individual neurons that you can then use, you can then combine to learn whatever task you're interested in. So it's kind of a very general framework for learning readouts, in fact, that perform certain tasks. So I believe in this setting, what's kind of cool about it is that the connectivity within the reservoir is left. It's just set so that the reservoir is stable, doesn't blow up, but there's lots of diverse activity coming in and out. So you don't learn that. What you learn is a readout for your specific task. So you can have kind of and interestingly, you can have different readouts for the same task kind of simultaneously. So you can have the same reservoir doing its thing, and you can have multiple readouts for different aspects of the task. So it's pretty cool. Okay, so we evaluated the computational capabilities of balance, so that's excitatory inhibitory balance, recurrent networks of rate neurons with neuron type specific connectivity patterns. So presumably the excitatory and inhibitory neurons will each have their own types of connectivity. Reservoirs are composed of several interconnected clusters of EI populations. Okay, interesting. And are trained to simultaneously predict the trajectories of multiple chaotic time series. Okay, so maybe that's interconnected clusters. So I wonder whether that's, so instead of having one big reservoir, it sounds like you have several smaller reservoirs that are interconnected and predicts the trajectories of multiple chaotic time series. So I wonder whether you're going to be doing that all at the same time, like I just mentioned. We studied the impact of varying E and I clustering levels on network dynamics and identified the optimal topology for a complex time series reconstruction task. The impact of varying E and I clustering levels. Okay, so I guess they're just trying to figure out, their goal is going to be, reproducing or capturing this chaotic time series, and they're going to be, I guess, changing the geometry of these clusters to see, what works best. We find that the presence of different levels of excitatory and inhibitory clustering enables a precise control of the network's dynamical state. In particular, okay, that doesn't seem super surprising. Enables a precise control. In particular, we show that the E and I clustering levels, distinct and non-trivial effects. Okay, so I'm not sure what they mean. E and I clustering levels, distinct, okay, so maybe they missed a word there, and non-trivial effects on network dynamics and identify structures that control the network's distance from chaotic state. Finally, we demonstrated that a commonly observed cortical continuity pattern of highly specific excitation and less specific but not uniform inhibition among similarly tuned neurons can maintain network dynamics close to the edge of chaos and may significantly contribute to the computational efficiency of brain networks. Okay, so this is this idea that you want your, I think it goes back to Stuart Kaufman actually, that if you have, and I'm kind of phrasing this very loosely, but if your system is too damped, if it's too stable, then you don't have, it's very slow to respond to new stimuli or to a time series you're capturing. On the other hand, if it's too excitable, then it becomes very sensitive to noise, and it also becomes kind of difficult to use to capture time series, do whatever task you want. So you want these systems to be on the edge, what they call this edge of chaos, where it's still very responsive. So it's it's got enough instability in it for it to be responsive to new inputs, but not so much that it just it's very sensitive, overly sensitive to noise, and it'll just blow up. So I think here, yeah. So here, I guess what they're trying to say is like, look, we do observe this in cortex. There is this, what they already mentioned, that there's similarly that high connection probability between similarly tuned neurons. So there is that between the excitatory neurons, and then there is this inhibition. But this inhibition they're going to model as not being uniform, and they're going to show that it has these very kind of interesting, useful properties that might be really beneficial if you're trying to, capture chaotic time series. You don't necessarily think that's what the brain is doing, but that could be a good stand-in for various tasks that different brain areas have to do. So I guess just looking at more realistic excitatory inhibitory motifs and the benefits it might bring to time series prediction and capture. Okay, so that seems pretty interesting. So that's the very first one. Okay, so let's see what else. Okay, so the next one is connectome-based models of feature selectivity in a cortical circuit. Okay, so let's see who that is. Let's have a look at that. Okay, so let's read this. So feature selectivity, neurons' heightened responses to specific configurations of stimuli, constitutes a fundamental building block of cortical functions. Okay, various mechanisms have been proposed to explain its origins, differing primarily in their assumptions about the connectivity between neurons. So I guess like one example of the feature selectivity would be like in V1 or the early visual areas where we think that maybe simple cells respond to these Gabor-shaped Gabor-shaped patterns. So that seems to capture some of what those cells are doing. Okay, some models describe selectivity as structured, tuning-dependent feed-forward or recurrent connections, whereas others have demonstrated that selectivity can emerge with randomly connected networks when interactions are sufficiently strong. This diversity of plausible explanations poses a challenge. So we're trying to figure out what's actually happening. So is it going to be structured, tuning-dependent connections, or are they just going to be random connections with, well, I guess, yeah, I'm not sure what they mean that it's sufficiently strong. We developed a novel approach that seeks to minimize preconceived assumptions about the underlying connectivity by utilizing connectomic data at synaptic resolutions to construct network models. Okay, so this was kind of one of the themes I thought would start showing up. So maybe they're gonna build a model using connectome data. So we're not going to say that connectivity is, for example, random, or it's set up according to what maybe the input tunings are, but we're actually just going to constrain it by what we actually know in a particular circuit. With this, we investigated mechanisms governing selectivity to oriented visual stimuli in mouse visual cortex. So this is kind of a classic stimulus set where you've got gratings moving in a particular direction, and you want to see how the neurons in different parts of the visual system are going to respond to that. We show that a connectome constrained network model mimics neural responses seen in experiments and points at randomness and connectivity as a dominant factor shaping selectivity in the circuit. Connectome constraint mimics neural responses. Okay, so presumably they're going to put in the connectivity they see and measure some properties of the circuit and show that, okay, look, it looks a lot like what we actually observe. I'm not sure what they mean, and points at randomness and connectivity as a dominant factor shaping selectivity in the circuit. I would have thought that randomness in connectivity, I wonder how they can assess that. Do they have multiple connectomes that they can say, well, look, between different animals, the connectivity seems random, but maybe the experience of the animals are different. These findings provide novel on mechanistic mechanisms underlying feature selectivity in cortex, and highlight the potential of connectome-based models for exploring the mechanistic underpinnings of brain function. So I think this will just be an interesting poster to see how they're going to use this connectome, how they're going to use the connectome to constrain their models and to compare it to real experiments. I think this is going to be definitely a trend in coming years as we get larger and larger connectomes, and ultimately that's kind of what we want to connect, right? The function of neural circuits to how they're built. So yeah, I think it'll be interesting just to capture that. I'm not so sure about some of the other points. I'm sure it'll get cleared up at the poster session. So I think that'll be a cool one to check out. Okay, so that's that. Let's have a look. Oh, this one seems interesting. So interpretable representations of neural dynamics using geometric deep learning. Okay, so this is again one of those themes I thought would come up, which is we've got these maybe deep networks that we can train to do tasks, but how are they solving it? So we have a black box that does the task for you, but how did it do it? So let's see what these guys are going to say. Okay, so increasingly recognize that computations in the brain and artificial neural networks can be understood as outputs of a high-dimensional dynamical system conformed by the activity of large neural populations. Okay, I mean, I think that's to some extent tautological, because that is what it is, right? It's the neurons. You have as many dimensions as neurons at least, and there's a time series. So you can think of that as a dynamical system. Yet revealing the structure of the underpinning latent dynamical processes from data and interpreting the relevance in computational tasks remains a fundamental challenge. Again, so we talked about this, right? So you're going to have the brain solving something or an RNN solving something, and it's going to solve it, and you want to understand that, and it will frequently turn out that high dimensional activity has a much lower kind of latent or intrinsic dimension, and so you're seeing kind of a high dimensional projection sort of a low dimensional dynamics. And that's kind of helpful because we have a chance of understanding those low dimensional dynamics. Just because the smaller number of dimensions, maybe it'll be more understandable. Okay, a prominent line of research has observed that task-relevant neural activity often takes place on low dimensional smooth subspaces of the state space called neural manifolds. So this is just what we talked about. However, there's a lack of theoretical frameworks for the unsupervised representation of neural dynamics that are interpretable based on behavioral variables, comparable across systems, and decodable to behavior with high accuracy. So that's interesting. So last year at Cosyne, the keynote was Zebra, where they provided a kind of data analytic framework for doing this kind of thing. So it had, it wasn't quite unsupervised. It was, let's say, self-supervised with the contrastive learning. But the point was that you could, the latent variables you got there were reproducible across animals. So you could compare, you were hoping you were kind of capturing something deep about what was happening rather than something that's very, animal specific. Okay, but these guys are gonna, I guess, look at this from a more theoretical perspective. Okay, to address these challenges, we introduced manifold representation basis learning. Okay, so we've got a nice acronym MARBLE, always important. A fully unsupervised representation learning framework for nonlinear dynamical systems. Okay. Our approach combines empirical dynamical modeling and geometric deep learning to transform neural activations during a set of trials into statistical distribution of local flow fields. Okay, so presumably you're going to see the high dimensional activity moving. You're going to see those points moving in a high dimensional space, maybe projected down to some manifold, and maybe you're going to learn the corresponding vector field that would underlie the flow that you observe. Maybe that's what they mean. Our central insight is that LFFs vary continuously over the neural manifold, allowing for unsupervised learning, and are preserved under different manifold embeddings, allowing the comparison of neural computations across networks and animals. So let's read that again. So it's that LFF, so local flow fields, vary continuously over the neural manifold. So I wonder, does that follow? If one tells you that the neural activity itself is continuous, well, not continuous, let's say smooth, so it's not making sudden jumps, and the neural activity is being determined by flow across, by these, let's say, these flow fields, these vector fields, does that already imply that the vector fields themselves are going to be smooth, varying smoothly? But I guess they're saying actually something weaker, so they continuously, so it's not necessarily smooth, depending on how they mean that. Allow and are preserved under different manifold embeddings, allowing the comparison of neural computation across networks and animals. So I guess what they want to say is that these manifold embeddings presumably would correspond to at least different animals solving the same task. So maybe the particular set of neurons solving tasks are going to vary from animal to animal, and you want the underlying structure that's captured to be kind of invariant to that. So maybe that's what they're talking about. So we show that MARBLE offers a well-defined similarity metric between different neural systems that is expressive enough to compare computations and detect fine-grained changes in dynamics due to task variables. Okay, so again, a lot to unpack here. So offers a well-defined similarity metric between different neural systems, maybe comparing these flow fields, expressive enough to compare computations and detect fine-grained changes in dynamics due to task variables. So maybe again, just looking at these flow fields perhaps. Being unsupervised, MARBLE is uniquely suited to biological discovery. Indeed, we show that it discovers more interpretable neural representation, several motor navigation than generative models such as LFADs, or semi-supervised models such as Zebra. Okay, interesting. So they're actually going to have not just the theoretical aspect, but some data, some tools for data analysis that they think are going to be better than LFADs and Zebra. Intriguingly, this interpretability implies significantly higher decoding performance than state of the art. Okay, so so I guess those two things don't have to go together, right? So you could, if you're just optimizing decoder performance, it's not interpretability might go out the window. There might be some, very complex projection of the data that actually gives you really good decoding. But they're saying that actually it turns out that the interpretability is going along with higher decoding performance. So yeah, a kind of a non-trivial coincidence. Our results suggest that using manifold structure yields a new class of algorithms with higher performance and the ability to assimilate data across experiments. So very interesting. Okay, so I think there's a lot to be understood there, but I think a key aspect of this is And we saw it last year. We see it, in our lab and other labs that you have these dynamics that you record across multiple animals, and you want to kind of make sure that whatever you're learning, you can compare and register across different animals. And it seems like they've got kind of a nice theoretical setup that's going to do that, and is competitive with the state of the art. So that'll be a fun one to see. I think a lot to have to understand of what they're up to. Nice. Okay, so let's continue. Okay, so interesting. Okay, so identifying the impact of local connectivity features on network dynamics. So that sounds really interesting. So yeah, so what's really cool about that is, again, so this goes down to the the connectome ideas. So if we change, if we observe a particular pattern of connectivity in a part of the circuit, how does that manifest in the dynamics? Or if we see certain types of dynamics, can we map it back to some connectivity features? So let's see what that sounds really exciting. Let's see what they've got to say. Okay, great. Okay, so understanding how connectivity structure shapes network dynamics is paramount in the field of neuroscience. Yep. Yeah, and this goes back to these themes that I was talking about, where we're looking at computation from, let's say, the algorithmic level, when we're looking at computational dynamics. So okay, so you might need a stable fixed point here where the dynamics converge to, and maybe on the way you need some particular manifold setup to make dynamics, go where it needs to go. But the question is now, the implementation level question is, well, how do you implement that in terms of connectivity and neural populations? So linking those is really important, and I guess that's the broad theme that these guys are going to be working in. Okay, so theoretical investigations of multi-population neural networks often consider statistically homogeneous populations and incorporate either only the population average mean or IID fluctuations in synaptic couplings. So often consider statistically homogeneous populations. So I guess what they're saying is, look, we're going to have a bunch of neurons whose activity statistically is going to be is the same, and we're just going to wire them up in a particular way and just see what the dynamics look like. But that's not what real neurons are like. Or IID fluctuation in synaptic couplings. So yeah, yeah, I guess we're going to change the synaptic strength, but we're going to assume they're all independent and identically distributed. So they're all the synapses are fluctuating all independently of each other and are kind of coming from the same distribution. So maybe Gaussian fluctuations. But again, not really anything like what the real brain, we think, is doing. Obviously, we have to do this first because it's already very difficult to analyze these systems, and we have to start somewhere. So it's important to consider those simple cases first before we take the step to more realistic networks. And I guess that's what they're doing here. Okay, a newly released synaptic physiology data set. Okay, interesting. So now bringing in, again, maybe not quite connectomics, but maybe the dynamics of conductances at these synapses, highlighted the strong presence of motifs. Okay, so yeah, this is actually connectomic stuff. Specific connectivity patterns between pairs and triplets of neurons beyond the scope of mean connectivity. However, it is a priori not clear which of the experimentally identified connectivity motifs exert a strong influence on neural dynamics. Okay, so we see certain patterns of connectivity, like maybe this population is presynaptic to that other population, but less so vice versa. How do these affect dynamics? While most previous works focused on reciprocal motifs, so when going there and back, here we show that another feature of connectivity, chain motifs, has a much stronger impact on the dynamics of neural activity. Okay, so that's cool. So, if you're looking at dynamics, that's going to be potentially determined by the presence of these chains where A connects to B, B connects to C, rather than these reciprocal motifs, which might be easier to analyze. We compare the effects of chain and reciprocal motifs within two population excitatory inhibitory networks, using an analytical framework that approximates the connectivity in terms of the low-rank structures that incorporate motifs. We mathematically derived the dominant eigenvalues and exploited matrix perturbation theory to determine the statistics of corresponding eigenvectors. We then use these results to perform a low-rank approximation that predicts the effects of connectivity motifs on linear network dynamics. Okay, so it's a lot to unpack here. So, okay, so we're gonna approximate connectivity in terms of lower rank structures that incorporates motifs. So connectivity can be, there's n by n, so that could be kind of a rank n matrix. They're going to first approximate it into a low rank in a low rank way, and somehow incorporating these motifs they're interested in. We mathematically derive the dominant and then exploit matrix perturbation theory to determine the statistics of corresponding eigenvectors. Okay. Right. So maybe they're going to start with the low rank structure, and then maybe put in motifs as perturbations. We then use the results to perform a low rank approximation that predicts the effects of connectivity motifs on linear network dynamics. So yeah, so perhaps, and we'll have to see, you have the low rank structure, and then you put in these motifs as perturbations, and then you do again a low rank approximation of that result, and then you see what a linear network with that connectivity, how it would behave. So yeah, that's an interesting idea. And our results show that chain motifs have a much stronger impact on dominant eigenmodes than reciprocal motifs. So that means, okay, so when you look at the activity in a linear network, that activity is going to be described in terms of these eigenvectors, these eigenmodes. And so what they're saying is that when we have the chain motifs have a much stronger effect on these modes. Moreover, an overrepresentation of chain motifs induces an additional eigenmode. So there's going to be an additional kind of population pattern of activity that will be inside the dynamics. Eigenvalue of sign opposite to the dominant one, thus modifying the network's effective rank. An additional eigenmode with an eigenvalue of sign opposite to modifying the network's effective rank. This additional eigenmode substantially influences network dynamics, offering a new perspective on how local EI motifs shape the network's excitability. Our exploration of the physiological connectivity data set for the first time revealed a significant impact of an EI chain motifs on altering the network's effective rank. What is the effective rank? I've seen some notion of this. I wonder if is it somehow related to the trace of the eigenvalues? So for example, now if you have, because it's not clear to me how, and I'm sure it will be once we see the poster, but how an additional eigenmode, so you would think an additional eigenmode would actually increase the rank, with an eigenvalue of sign opposite to the dominant one. So in principle, so if you have a third eigenmode, third eigenvector with an opposite sign, that should be an increase in rank. But if you're maybe the effective rank in these dynamics is maybe related to the trace, and then in that case, maybe it changes. That will change the effective rank, have some interesting effects. Exploration of physiological connectivity data set for the first time revealed a significant impact of this chain motif on altering the network's effective rank, discovering richer dynamics associated with these specific connectivity motifs. So that's very cool. So I think that's a really nice, sounds like a really nice exploration of taking again some connectomic data, looking at, okay, some motifs are there. How are we going to understand the effect of these motifs on the dynamics? Well, of course, we have to make some approximations, and these low-rank approximations have been shown to be quite effective in capturing neural activity, how neural systems solve particular tasks. And so yeah, so they've been able to use kind of similar ideas to look at the effects of these motifs and come up with a really interesting, new aspect of the connectivity to focus on these motifs. So yeah, I think that'll be a really good one to chat about. Nice, great. So let's go and have a look at the next one. Okay, so let's see, an olfaction one. Excellent. Okay, so when is a non-canonical olfactory system optimal? What is this about? Okay, so let's see. The early olfactory system is classically described by canonical one receptor, one neuron, one glomerulus organization. Yeah, so this is kind of one thing we always say. The receptor the receptors are spread over the epithelium, but they all go based on what receptor they're expressing to the same glomerulus. And the one neuron thing is that every OSN, olfactory sensory neuron, expresses one receptor. So one receptor being expressed in one OSN, which all go to the same glomerulus. Yep, that's exactly what they say. This model roughly describes the early olfactory system of Drosophila, the mouse, humans, other species. It's however not universal. The mosquito, Aedes aegypti, expresses at least twice as many ORs as it has glomeruli. Twice as many ORs. Okay, so interesting. So where are they going? So each glomerulus must pool inputs driven by multiple ORs. Herrera et al. Found that individual Aedes aegypti ORNs co-express multiple ORs. Interesting. So you have each neuron is expressing multiple ORs, and they're going to a smaller number of glomeruli than there are different receptor types. So I mean, one way this could have been set up would be, imagine, if you had, let's say each ORN expressed 2, then you would think, okay, well, there's going to be roughly n squared now combinations of the 2 ORs, and so there's going to be n squared different glomeruli if it was kind of one OR by if the receptors you expressed determined where you went. But it actually looks like they express 2, and they converge to fewer glomeruli. So that's really interesting. Why does violate the canonical model of the earlier? We take a normative approach, assuming that the glomeruli inputs encode olfactory stimuli in a noisy environment. Okay, very valid. We analytically compute the patterns of receptor expression which maximize the mutual information between olfactory stimuli and glomerular inputs under a resource constraint for OR expression. We find that pooling ORs for correlated odorants is optimal when the olfactory signal is weak or the olfactory environment is noisy relative to neural responses. I see. Okay, so that goes to the classic result. The top principal components maximize mutual information in Gaussian environments. On the other hand, selecting one high variance receptor in each glomerulus when the olfactory signal is strong or neural responses are unreliable. By training feed-forward networks to perform an odor classification task, we show that this pattern persists in the presence of response nonlinearities. Confirming our predictions, Drosophila melanogaster ORNs co-express 2 receptors, OR33C and OR85E, whose ligands are correlated across natural stimuli. Our work suggests that the organization of early olfactory systems reflects the statistics of the ethologically relevant olfactory environments experienced by different species. Okay, so I think the idea there is, so the idea is that if you have, there's the concept of efficient coding, which says that, okay, if the noise levels are low, then what you want is the output of your different, like your receptor channels to be, let's say, receptor channels to be decorrelated, so that each of them kind of gives you different information about the environment in a low noise setting. But in a higher noise setting, you actually might want to keep those correlations because they will reinforce and allow you to extract the signal. So I think what they're saying here is something like that is happening in the Aedes aegypti ORN. So if you're in kind of a particularly noisy environment, maybe you might want to start co-expressing ORs that are responsive to kind of very similar ligands to allow you the output of that neuron to essentially allow to communicate something about that part of olfactory space in the presence of noise. So get the 2 different types of activation, have them excite the same neuron, pool those results, and allow you to pull out the signal. Now it's interesting because they're also imposing. There seems to be 2 things going on here. There is the energy constraint and the noise. So the energy constraint on, but it's not the energy constraint on activation. A resource constraint for OR expression. So that's quite different. So like in the efficient coding setting, the energy constraint is like on the output of your units. Here it's like how many channels of input do you have? And I wonder, you would expect, even without such a constraint, you might expect that you would want this pooling of correlated activity. So I wonder what what this resource constraint contributes, if you actually even need it. But I guess we'll find out. Yeah, and also, yeah, because they're saying, look, it's happening kind of a noisy setting. I mean, it's also noisy. I mean, the olfactory environment is kind of noisy. I would assume equally for flies and for, let's say, for humans and mice. But maybe in the flies, because you just have a smaller number of receptors, that signal that you're getting is maybe weaker. So maybe you need to kind of play these kinds of tricks to pull out the signal that you need to pull out the signal from the noise. Interesting. Anyway, that'll be a fun one to have a look at. Okay, let's see. Okay, here we go. So actually, I've only seen one of these, one abstract so far, talking about large language models. So I think my predictions on that front so far have been very poor, but let's see. Okay, so what are they going to be talking about? Right, so language encoding models predict neural activity during language comprehension tasks from stimulus representations. State-of-the-art performance in language encoding uses learned representations from LLMs, suggesting similarities between how LLMs and humans process language. However, little progress has been made in the interpretation of these predictive features. Okay, so learned representations from LLMs suggest a similarity between how LLMs and humans process language. However, little has been interpretation of these features. So again, we're kind of in that same boat, interpreting what these neural networks are doing. Here we sought to understand how much of the linearly mappable information between LLMs and brains is accounted for by explicitly interpretable feature spaces. Towards this end, we collected a set of interpretable feature spaces that we hypothesized would be useful for language encoding. These consisted of positional signals tracking the comprehender's location in the passage of text, static semantic embeddings derived from glove, and 3 surprisal values computed using an LLM. We then examined what fraction of LLM neural predictivity can be explained by these interpretable feature spaces through a variance partitioning approach. We applied our method to an fMRI dataset in which participants read short passages where both untrained and trained GPT-2 large models were reported to display high neural predictivity in an influential prior study. We found that 1, positional signals and random word embeddings fully account for untrained GPT-2 neural predictivity, and that 2, a combination of positional, semantic, and surprisal feature spaces accounted for 80% of the neural variance. Additionally, GPT did not explain significantly more neural variance in total than the interpretable feature spaces. Together, these results suggest that the majority of neural variance explained by GPT-2 on a passage-level data set can be accounted for by a concise set of interpretable feature spaces. Okay, so yeah, so this isn't quite my corner of the field, but I guess they're trying to say, well, how do we explain what these LLMs are doing? And so they've picked some features that they think are going to be useful, and they find that you can kind of capture a good chunk of the variance that's explained by GPT-2, which is admittedly an earlier version of these GPT models. So I don't know how it would apply to the later models by these interpretable feature spaces. So it's a case of kind of guessing what those features are and then comparing to the network. Okay. All right. So let's see. Okay. So this is interesting. Let's have a look at this. See more. All right. I'm sorry. What was it called? Riemannian geometry of neural object representations. Okay. Sounds really cool. So what are they going to be talking about? Okay. So neural codes are distributed. The identity of a particular stimulus is usually signaled not by the activity of a single specialized neuron, but by collective change in the activity of a population. Okay, yep. So this has been the big realization for the last about 20 years or so, that we need to look at what's happening on a population level to understand what a neural circuit is doing. Understand distributed neural codes, we need to identify the right set of collective observables one should measure. Okay. So we have, neural activity of thousands of neurons. What summaries of that activity should we look at to understand what it's doing? So, and other works look at this in terms of like those low-dimensional latent variables. Here we propose that the Riemannian geometry of neural representations provides a compelling candidate framework. Okay, so we'll have to see what they mean by that. This perspective gives a precise way to measure how distributed codes shape the similarity of stimuli. It is particularly powerful because it does not require a priori knowledge of the geometric structure for which one is looking. As a proof of principle, we investigate the geometry of deep convolutional neural networks trained to perform object classification, okay, which are a popular model for computation in the ventral visual stream. So that's the object detection path. We show that these networks learn to magnify areas of the input space around object class boundaries. Okay, so that's kind of where the Riemannian stuff comes in. So you've got a metric that varies with your location, on the manifold, and I guess they're saying that at those boundaries, the metric is perhaps expanded in particular directions maybe perpendicular to the boundary, along the boundary, whatever it is. Networks trained with self-supervised learning also learn to magnify areas near decision boundaries. This provides an important prerequisite for applying these analyses to biological data. Even when training is performed without label information, object information can be extracted from the geometry of neural representation. That's really cool. This provides an important for applying, even when training is performed without label information, object can be extracted from the geometry of the neural representation. So if you look at how that, I guess this, the metric of that, the Riemannian geometry, if you look at the right properties of that, looking for these expansions, you might already get a sense of, maybe what the categories the system is already trying to distinguish. This leads us to hypothesize the dimensional visual stream may share this object-centric geometric structure. In total, this Riemannian framework provides a new lens through which to compare natural and artificial distributed neural representations. That sounds really interesting. Riemannian geometry. Yeah, so that's okay. Compelling count. Yeah, that'll be cool. I'll have to see what it's all about, but let's have a look.